Now we can talk, yesterday we talk a lot about the bacteriophage and history, evolution, how it can be used. So now today we will come with the topic where the bacteriophage, especially we talk about the lambda. So this is the name of the bacteriophage that can infect the Escherichia coli bacteria. And this bacteriophage have the either lytic cycle or lysogenic cycle. So in the lytic cycle, they are going to lyse the bacteria, and the lysogenic cycle, they are going to integrate their genome into the bacterial, and they are going to be still there until or unless induction is not going to happen. They are remain there. Once they are going to induce, it can do the lysis. So what are the bacteriophage and how it reproduce? So bacteriophage exists by the invading or infecting and manipulating the bacterial host cell. So the bacteriophage can act and attack on the bacteria. Then they can release their DNA and they can take the host machineries, and with the taking the host machineries they can propagate, yeah, it can reproduce and produce a new phage. And this new phage is only possible because they are taking the host machineries. So phage independently cannot do anything until or unless they are not going to attack or infect the bacteria. Once they infect the bacteria, specific bacteria, they can take the whole machineries like the all the transcription, translation, even DNA replication, whole machineries they are taking from the bacteria, and they are going to multiply, yeah, propagate. So what are the characteristic feature of the phage genome present in the phage particle, and what is the characteristic of the phage genome in the host cell? So there are the differences. So bacteriophage, here we talk about the lambda. So it has the linear genome, double helix, double stranded, and enclosed with the protein body. So the bacteriophage have the double helix linear DNA that is enclosed with the protein body. And the bacteriophage have the three part. One is the head where the it has the DNA, and they have the stall. So it's long stall, and they have the tail. So these are the three components present in the lambda phage. And it has the linear genomic DNA, but this linear genomic DNA have the sticky overhang. Sticky overhang means they have the compatible site and they are easily aligned. So when it is present in the host, it is present in the phage, they have the linear double helix DNA. But if it's transfer, this phage, if transfer this genome into the host, then they become the circular. So this sticky end are the complementary to each other, so it is easy to make the joining. And when the linear DNA of the phage is injected into the host cell, the sticky end complement each other to make the circular. So it is very easy to make this linear to the circular. So there are the two conditions, they make this linear DNA become the circular DNA. So the life cycle of the lambda bacteriophage, they process the two step, two form. One is the lytic, another is the lysogenic. So in the lytic cycle, the phage genome is replicated into the many copy, and they are taking the whole machineries of the bacterial system, and they make the DNA replication, transcription, and the translation. And this transcription, translation make the, and they are going to assemble. So all this protein molecule, nucleic acid molecule, they are going to assemble, and they can produce the new progeny, and this is going to gets released from the host by lysis or destroying, and they can infect the new bacteria. So this is the like the co-infection, it is going to happen during the lytic cycle. So the second form or life cycle of the phage is they have the genome, but it is not replicated. The phage genome is not replicated, and the phage genome is integrated into the host genome. So normally the phage, the transfer the DNA into the host, so they are not going to replicate, they are not going to do anything. They have the integration inside the host genome, and because of the integration in the host genome, they are not producing any progeny. So when the host cell is not destroyed, the replication of the phage genome achieve when the bacterial cell is replicated. So for example, if one bacteria divides into the two, the whole genome is going to make this one genome to two genome. In that time, only the phage genome is replicated. So this is the cycle where in the first step, this is the lytic cycle and the lysogenic cycle. In the lytic cycle, you can see the phage, it is going to attach on the surface of the bacteria, and the bacteria have also own genome. So it's blue color. So once the entry of the bacteriophage genome into the cell, they are the cell in the, they are going to synthesize and taking the machineries from the bacterial cell, and they can do the replication, they can do the transcription, translation, and all the components are assembled together and they can make the new page. And once the page number is like 500 to 600 going inside the bacterial cell, they are going to lyse the bacteria, and the phage is going to release outside and they can infect similarly in the same manner. So this cycle is repeated. So that is kinds of the lytic cycle phage have. Another cycle is called the lysogenic cycle. So in the lysogenic cycle, so the step first, this is the attachment of the phage on the surface of the bacteria, and they can release the DNA into the bacterial cell, and they this release of the DNA bacterial cell, it is going to integrate into the genome of the bacteria, and that is called the prophase in the chromosome. So once the bacteria is divided, the genome of the bacteria as well as the genome of the virus, the bacteriophage, it is going to replicate it. So it is happen in the only the case of the division of the cell. So they are not going to make any kinds of the bacteriophage, progeny, bacteriophage inside there. So in the cell division, it's only happen and the bacteria is going to divide. So certain condition like the induction, if the bacteria is get like the UV induction, chemical induction, then in that case the genome of the phage, it is going to out from the genome of the bacteria, and they are going to do the replication and the transcription, translation, and they can make the, they are going to assemble and it can follow the cycle like lytic cycle. So they can release the page and they are going to, so this is the two condition is present in the lambda phage life cycle. So when you use to each of the cycle, what are the regulates the choice? So that is called the molecular switch. So in which condition the phage is going to take the lysogenic or the lytic cycle? It has the molecular mechanism inside. So you will see here, this is the circular DNA, circular genome of the bacteriophage, and this have the two orientation transcription. One is the left orientation transcription, another is the right orientation transcription, right side. So they are follow the left and right. So you can see there is the blue part that is called the cos site, and the cos site is the sticky in the site present on the genome of the bacteriophage, and they are compatible. So once the phage is released into the bacteria, this cos site is going to join because of the complementarity present in the this region, and they are doing the transcription. they can take place either the left direction or the right direction. So you will see here the yellow location in the genome where it is represented the operator and the promoter and the transcription site. So PL is the one promoter, PR is the another promoter, and they have the O, one operator for the PL promoter, so is OL, and another is for the R, so it is PR, so it is operator for R, so OR. And the lambda C1, this is the transcription factor, and they are used the role as the repressor. So if you look the inside, they how they are going to either follow the lytic or lysogenic cycle. So it has the control. So once the transcription is left side, turn the gene to the integrate the phase genome into the cell. So turning up and on in the yellow side, in either right side or left side, it can be decided by here. So there are the switch that is called the molecular switch, and it is based on the lambda repressor protein, that is lambda C1. So it can activate the lysogenic cycle. So the lambda repressor, they can activate the lysogenic cycle. There is the another protein that is called the CRO protein. So if this CRO protein, it can activate the lytic cycle. So this system is called lambda repressor switch because its activation is going to happen based on this protein. So this lambda C1, they have the binding site on the DNA, and you can see here the atomic level structure of the lambda protein with the DNA. So what happened, because the yellow site, it can be magnified here, and you can see easily there is the three promoter. One promoter is the PL promoter, second promoter is the P lambda C1, third promoter is the PR. So the P lambda C1 promoter express the C1 protein. So this is the lambda C1, yeah, you can say the C1 protein. And the PR promoter, they can express the CRO protein. So PR promoter for the transcribing the right side of the gene, PL promoter promote the transcription of the left side of the gene, PC1 promoter that can transcribe the C1, lambda C1 repressor. So these are the three important promoter. They can play the important role for the either lysogenic cycle is going to happen, or lytic cycle is going to happen, and how they are going to regulate. So for example, if the lytic cycle is achieved when both promoter PR and PL are turned on, but but in that case the P lambda C1 is turned off. So if the P lambda C1 is turned off, so the bacteriophage can follow the lytic cycle. So if lysogenic cycle is achieved when the both PC1 is turned on and the PL and the PR turn off. So if only the lambda C1 is turned on, so it can express the lambda C1 repressor. In that condition, the bacteriophage can follow the lysogenic cycle. So here you can see in the region how the lambda C1 and CRO gene that include the PC1 and the PR promoter. There are the DNA motif that is called the operator region present there. So operator is playing the important role where the transcription factor or repression can bind there. Either they can activate the gene function or they can repress the gene function. So in this condition, you can see operator region in the bacteriophage genome, it is overlapping with the promoter region. So you can see here they are going to overlap some of the part. So when the CRO is expressed, when the CRO gene is expressed, it can produce the CRO protein. So you can see the orange color, they can produce the more CRO protein. And this CRO protein, it can bind with the promoters lambda C1. So the CRO protein bind to the operator region of the overlapping promoter lambda C1. So they can repress the gene function of the lambda C1. And the CRO also prevent the RNA polymerase. They cannot bind with the promoters C1. So because of the inhibition. So if the CRO protein already bind on the operator side of the PC1, then it cannot allow, and there is the competition. So it cannot allow the RNA polymerase can bind there. So in that condition, there is no lambda C1 protein produced. So in this condition, as a result, lytic cycle gets gene expressed because of the activity of the promoter. So if the lambda C1 is not active, in that condition, there is no expression of the lambda C1 repressor. And in this case, the both the promoter PL and PR is active, and the bacteriophage follow the lytic cycle. So if C1 is expressed from the C1 protein, C1 is expressed, C1 gene is expressed, and it can produce the C1 protein. So the C1 can bind to the operator region of the overlapping CRO gene and other gene of the PR. So they can repress, and the C1 that can prevent the polymerization of the binding site of the PR promoter. So there is no CRO protein. So there is no lytic pathways is going to produce. So in this condition, as a result, lysogenic pathways gene are expressed. So in this condition, say C1 protein bind to the operator region of the R1 and R2, and cooperatively it can help for this lysogenic cycle. So this is the kinds of the cooperation and the cooperative binding. So it is always happen with the cooperation of the binding and the affinity of the interaction, and the concentration of both the protein, both the repressor protein is very, very important role. Either the bacteriophage follow the lytic cycle or lysogenic cycle. So that is called kinds of the molecular switch. So in this condition here, we can see the lambda C1 is expressed and it can produce the C1 protein. So all promoter are blocked. So no further C1 is produced. So if there is no further C1 is produced by the cooperative binding, and it can express an own expression and slow down the occupying PC1 promoter region, and it can stop the activity. So here you can see as a summary, lambda repressor, they can bind on the region of the operator, and after the induction, the pro-protein, they are going to be activated and they can move. So it can be kinds of the molecular switch, whether lytic cycle or a lysogenic cycle, it is going to happen and they are going to make the cycle complete.